A membrane is said to be at its resting membrane potential when it has a membrane potential of negative 70 megavolts. The resting membrane potential is maintained by the sodium-potassium pump, which pumps sodium out and potassium in using ATP. The sodium is pumped out 3 at a time and the potassium is pumped in 2 at a time. Both potassium and sodium are positively charged ions, but the ratio at which each is moved through the membrane creates a gradient in which the outside of the membrane is positively charged in comparison to the inside. At this time, there are also open channels to let both ions diffuse in and out, but there are more potassium channels. A receptor located at the dendrites receives a neurotransmitter from another neuron. The neurotransmitter binds the receptor and activates a G protein that opens a sodium-gated channel. The sodium ions diffuse throughout the cell, creating an action potential and changing the membrane potential to negative 55 megavolts. As a result, the voltage-gated channels open, letting sodium diffuse in. The sodium continues to diffuse down the axon, moving towards the terminal end and triggering more voltage-gated channels. The opening of voltage-gated channels along the axon propagates the action potential. Once the charge of the area is positive 35 megavolts, the sodium channels close and potassium gates open. Then potassium floods out, bringing the membrane potential down. These gates close once the charge is negative 80 megavolts, preventing another action potential from accidentally starting. The sodium-potassium pump then brings it back to negative 70 megavolts. The action potential reaches the axon terminal and causes a calcium-gated channel to open. Calcium diffuses in and binds to snare proteins. The snare proteins bind to each other and bring the vesicle near the membrane. The vesicle fuses with the membrane, and then the neurotransmitters inside are released by exocytosis. A receptor located at the dendrites receives a neurotransmitter from another neuron. The neurotransmitter binds the receptor and activates a G protein that opens a sodium-gated channel.